Good afternoon, class. Today we're going to look at Unit 12, Chapter 19, Part B, the cardiovascular system, blood vessels. Section 19.8, regulation of blood pressure. So this is a continuation of Part A. Let's start with homeostatic imbalances in blood pressure. Transient elevations in blood pressure occur during changes in posture, physical exertion, emotional upset or fever, and these are normal changes, adjustments to the blood pressure. Also, age, sex, weight, race, mood, and posture may affect the blood pressure. Is a sustained elevated arterial blood pressure of 140 over 90 millimeters of mercury or higher. Pre-hypertension is if values are elevated but not yet in this hypertensive state range. It may be transient adaptations during fever, physical exertion, and emotional upset, but it's often persistent in obese people. Hypertension can be dangerous because it is a major cause of heart failure, vascular disease, renal failure, and stroke, and it causes the heart to work harder, and the myocardium enlarges, weakens, and becomes flabby, and it also accelerates atherosclerosis. There's primary hypertension and secondary hypertension. So 90% of hypertensive conditions are primary. There's no underlying cause that can be identified, but there are some risk factors which include heredity, diet, obesity, age, diabetes mellitus, stress, and smoking. There's no cure, but it can be controlled with a restrictive diet where we restrict salt, fat, and cholesterol. Also increase exercise, lose weight, and stop smoking. And there are antihypertensive drugs. Hypertensive is less common, and it's due to identifiable disorders including obstructed renal arteries, kidney disease, and endocrine disorders such as hyperthyroidism and Cushing's syndrome, and treatment really focusing on correcting the underlying cause. Hypotension, so hypo means less, so it's low blood pressure below 60 millimeter of mercury, usually not a concern unless it causes inadequate blood flow to tissues, and it's often associated with long life and a lack of cardiovascular illness. Orthostatic hypotension is a temporarily low blood pressure and dizziness when suddenly rising from sitting or a reclining position. Chronic hypotension is a hint of poor nutrition and warning sign for Addison's disease or hypothyroidism, and acute hypotension is an important sign of circulatory shock. Circulatory shock is a condition where blood vessels inadequately fill and cannot circulate the blood normally. Inadequate blood flow cannot meet the tissue needs, basically. Hypovolemic shock results from when there's a large-scale blood loss. Vascular shock results from extreme vasodilation and decreased peripheral resistance. And cardiogenic shock results when an inefficient heart cannot sustain adequate circulation. Section 19.9, control of blood flow. So tissue perfusion is the blood flow through the body tissues, and it's involved in delivery of oxygen and nutrients to and removal of waste from the tissue cells. It also includes gas exchange at the level of the lungs, absorption of nutrients in the digestive tract, and urine formation in the kidneys. The rate of flow is precisely the right amount to provide proper function to that tissue or organ. Blood flow is controlled by extrinsic and intrinsic factors. Extrinsic factors involve the sympathetic nervous system and hormones that control blood flow through the whole body, that act on the arteriolar smooth muscle to reduce the flow to regions that need it the least. Control involves autoregulation at the local level to control blood flow. Blood flow is adjusted locally to meet specific tissues requirements. So it's the local arterioles that feed the capillaries and can undergo modification of their diameters, and organs regulate their own blood flow by varying the resistance of their own arterioles. Figure 19.14 is a quick summary of intrinsic versus extrinsic control mechanisms. So intrinsic controls is the control which is entirely from within the tissue or organ, so uses paracrines or locally acting hormones or properties of the muscle tissue, and it's also known as autoregulation or local control. Extrinsic controls instead is the control is coming from outside of that tissue or organ, and it uses nerves or hormones. So examples where both control mechanisms occur are the stroke volume in the heart that we've already seen, arteriolar diameter, which we're going to see, and glomerular filtration in the kidneys. This is in chapter 25, which we'll see next semester. So an example is redistribution of blood during exercise. So at rest, skeletal muscles receive about 20% of the total blood in the body, but during exercise, skeletal muscle can receive over 70% of the blood. So intrinsic controls, so at the level of the skeletal muscle, the arterioles dilate, increasing blood flow to the muscle, whereas extrinsic controls decrease the blood flow to other organs such as kidneys and digestive organs, and the mean arterial pressure is maintained despite dilation of skeletal muscle arterioles. 1915 shows the distribution of blood flow at rest and during strenuous exercise. So notice that at rest, skeletal muscle will only receive about 1,200 milliliters per minute of blood, whereas during exercise it will receive about 12,500. Let's look at intrinsic autoregulation of blood flow. Regulation of blood flow. So autoregulation are local or intrinsic conditions that regulate blood flow to that area. Reactive hyperemia is an increased blood flow to an area due to intrinsic factors. Two types of intrinsic mechanisms both determine the final autoregulatory response. Those are metabolic controls and myogenic controls. Myogenic involves muscles. So metabolic controls, increase in tissue metabolic activities results in the declining levels of oxygen and increasing levels of metabolic products such as H+, K+, adenosine, and prostaglandins. Effects of change in levels of local chemicals cause a direct relaxation of arterioles and relaxation of precapillary sphincters, and it causes the release of nitric oxide, which is a powerful vasodilator, by the endothelial cells. Endothelians also released from the endothelium are potent vasoconstrictors. Nitric oxide and endothelians are usually balanced unless blood flow is inadequate, in which case the nitric oxide wins control, causing vasodilation. Inflammatory chemicals can also cause vasodilation. Look at myogenic control. So myogenic responses are local vascular smooth muscle. It's when local vascular smooth muscle responds to changes in the mean arterial pressure to keep perfusion constant to avoid any damage to the tissue. Passive stretch is an increased mean arterial pressure that stretches the vessel wall more than normal. The smooth muscle responds by constricting, causing a decreased blood flow to the tissue. Reduced stretch is when there's a decreased mean arterial pressure that causes less stretch than normal, and in response, the smooth muscle dilates, causing an increased blood flow to the tissue. Long-term autoregulation. This occurs when short-term autoregulation cannot meet the tissue nutrient requirements. So long-term autoregulation steps in, but it may take weeks or months to increase the blood supply. So number of vessels to the region will increase. This is called angiogenesis, and existing vessels enlarge. And it's common in the heart in heart when coronary vessel occluded, or throughout the body in people in high altitude areas. Or actually, it also is something that happens during pregnancy. 1916, intrinsic and extrinsic control of arteriolar smooth muscle in the systemic circulation. Okay, so we have intrinsic controls, or autoregulation, in light green, I guess, and extrinsic controls, this is in orange. So vasodilators, these are going to be in so the metabolic intrinsic controls response to low oxygen, high CO2, high H+, high K+, prostaglandins, adenosine, and nitric oxide. And neural controls would be a decrease in the sympathetic tone and the hormone release of atrial natriuretic peptide, that is a hormone that is produced by the atria of the heart. In terms of vasoconstrictors, myogenic for stretch, so at the level of the muscles, metabolic with the release of endothelins, and neural controls would be increasing the sympathetic tone, and hormonal will be increasing angiotensin II, antidiuretic hormone from the posterior pituitary, epinephrine, and norepinephrine from the adrenal medulla. Flow in special areas. So let's look at the level of the skeletal muscle. So blood flow varies with fiber type and activity. At rest, myogenic and neural mechanisms predominate and maintain the flow at at approximately 1 liter per minute. Active or exercise hyperemia. During muscle activity, blood flow increases in direct proportion to metabolic activity, and local controls override the sympathetic vasoconstriction, and flow can increase tenfold. Figure 1917, active hyperemia. So exercising skeletal muscle is going to have an effect of a decreased oxygen level, increased CO2, and increased H levels, and other increase in other metabolic factors in the extracellular fluid. This is going to have the effect of vasodilation of arterioles that overrides extrinsic sympathetic input and increase the muscle blood flow in active hyperemia. Another special area is the brain. So blood flow to the brain must be constant because neurons are intolerant of ischemia. Flow averages are about 750 mils per minute. So the control mechanisms are metabolic controls. So a decreased pH or an increased CO2 causes marked vasodilation. Very high CO2 levels depress the autoregulatory mechanisms. Control mechanisms, so myogenic controls, a decreased mean arterial pressure causes the cerebral vessels to dilate, and increased mean arterial pressure causes cerebral vessels to constrict. The brain is vulnerable under extreme systemic pressure changes, and a MAP below 60 millimeters of mercury can cause syncope or fainting. Whereas a MAP above 160 millimeters of mercury can result in cerebral edema. Skin is another special area. So functions of blood flow through the skin supplies nutrients to the cells. It's autoregulated in response to oxygen needs. Also helps regulate the body temperature, which is neurally controlled, and it's an important function of skin. And it also provides a blood reservoir, which is also neurally controlled. Blood flow through venous plexuses below the skin surface regulates body temperature. The flow varies from 50 milliliters per minute to 2,500 milliliters per minute, depending on body temperature. Flow is controlled by the sympathetic nervous system reflexes, and reflexes are initiated by temperature receptors and the central nervous system. As the temperature rises from heat exposure, fever, or vigorous exercise, the hypothalamus signals to reduce vasomotor stimulation of skin vessels, causing dilation. The warm blood flushes into the capillary beds, and heat radiates from the skin. As temperature decreases, the blood is shunted to deeper, more vital organs. Superficial skin vessels constrict strongly, and blood in the vessels may become trapped, and it causes the rosy cheeks in the cold. Lungs, the pulmonary circuit is unusual and the pathway is short. Arteries and arterioles are more like veins or venules because they are thin-walled and they have large lumens. Arterial resistance and pressure are much lower than in systemic circuit, and it averages 24 over 10 millimeters of mercury versus the 120 over 80 millimeters of mercury in the systemic circuit. Autoregulatory mechanisms are opposite. Low oxygen levels cause vasoconstriction, and high levels promote vasodilation, and it allows the blood flow to the oxygen-rich areas of the lungs. At the level of the heart, the blood flow through the heart is influenced by aortic pressures and ventricular pumping. During ventricular systole, coronary vessels are compressed, and myocardial blood flow ceases, and stored myoglobin supplies sufficient oxygen. During diastole, high aortic pressure forces the blood through coronary circulation. Coronary blood flow is approximately 250 mL per minute, and the control is probably via myogenic mechanisms. During strenuous exercise, the coronary vessels dilate in response to local accumulation of vasodilators. Blood flow may increase 3 to 4 times, and it's important because the cardiac cells use 65% of the oxygen that is delivered. The other cells use only 25% of the delivered oxygen. Increasing coronary blood flow is the only way to provide more oxygen. Section 1910, capillary exchange. Velocity of blood flow. Velocity of flow changes as the blood travels through systemic circulation. It's fastest in the aorta and slowest in the capillaries, and then it increases again in the veins. Speed is inversely related to the total cross-sectional area. So capillaries have the largest area, so it's got the slowest flow, and the slow capillary flow allows adequate time for exchange between the blood and tissues. 18, blood flow velocity and total cross-sectional area of vessels. So we can see that the velocity of blood flow is inversely proportional to the total area of the vascular bed. So in the aorta, that has the lowest total area, it has the greatest velocity of blood flow, as opposed to the capillaries that have the highest total area, highest total cross-sectional area, and however the velocity is much slower. Vasomotion. So vasomotion is the intermittent flow of blood through the capillaries, and it's due to the on-off opening and closing of the precapillary sphincters. Capillary exchange of respiratory gases and nutrients. So many molecules pass by diffusion between blood and the interstitial fluid, and they move down their concentration gradients. That means from a high concentration to a low concentration. And molecules use 4 different routes to cross the capillary. The first one is they diffuse directly through the endothelial membranes, and the example are lipid-soluble molecules such as the respiratory gases. Another route is they pass through clefts, and this, for example, water-soluble solutes. Another route is they pass through the fenestrations. An example are the water-soluble solutes as well. And the fourth way is through active transport via pinocytotic vesicles or caveolae. And this is for the larger molecules such as proteins. Figure 1919 shows the capillary transport mechanism. So we see on top the pinocytotic vesicles. We see through the endothelial cell the fenestrations or pores, and the intercellular clefts. So figure 1919, capillary transport mechanisms. Let's start at one. So diffusion through plasma membrane, that is, again, in summary, we're summarizing the lipid-soluble substances. 2 is through movement through the intercellular clefts for water-soluble substances. 3rd, another way, is through the movement through fenestrations. Again, this is water-soluble substances. And 4, transport via vesicles. This is movement of large substances. Fluid movements, bulk flow. So fluid is forced out the clefts of capillaries at the arterial end, and most returns to the blood at the venous end. This is extremely important in determining the relative fluid volumes in blood and the interstitial space. Bulk flow, fluid flow across the capillary walls causes a continuous mixing of fluid between the plasma and the interstitial fluid, which maintains this interstitial environment. The direction and the amount of flow depend on 2 opposing forces, the hydrostatic pressures and the colloid osmotic pressures. The hydrostatic pressures are the hydrostatic pressure, HP. This is the force exerted by the fluid pressing against the wall. There are 2 types. The capillary hydrostatic pressure, or HPC, is the capillary blood pressure that tends to force fluids through the capillary walls. It's greater at the arterial end, about 35 millimeters of mercury of the bed, than at the venule end, which is around 17 millimeters of mercury. And the other hydrostatic pressure is the interstitial fluid hydrostatic pressure, or HPIF. This pressure is the pressure pushing fluid back into the vessel, and it's usually assumed to be 0 because lymphatic vessels drain the interstitial fluid. The colloid osmotic pressures, or these are the capillary colloid osmotic pressure, for example, or oncotic pressure. This is the sucking pressure created by non-diffusible plasma proteins that are pulling the water back into the capillary. Okay, so these are found in, those plasma proteins that are found within the plasma that draw the fluid back in, and it's around 26 millimeters of mercury. There's also the interstitial fluid colloid osmotic pressure. This pressure is inconsequential because interstitial fluid has very low protein content, and it contributes only about 1 millimeter of mercury. So the interaction of the 2, the hydrostatic and osmotic pressures, so the net filtration pressure comprises all forces acting on the capillary bed. So that would be the hydrostatic pressures minus HPC plus OPIF, interstitial pressure, minus the HP interstitial pressure plus the oncotic pressure, and the net fluid flow out of the arterial end, that's called filtration, whereas net fluid flow in at the venous end, that's called reabsorption. So more fluid leaves at the arterial end than is returned at the venous end. Excess interstitial fluid is returned to the blood via the lymphatic system. So anything that doesn't get back in, the lymphatic system will drain it. 19.1, bulk flow across the capillary walls. So each day, 20 liters of fluid filters from the capillaries at their arteriolar end and flows through the interstitial space. Most, about 17 liters, is reabsorbed at the venous end, and about 3 liters per day of fluid and any leaked proteins are removed by the lymphatic system. You can see the lymphatic capillary in green. And so where does the fluid go? It moves through the interstitial space. So let's look at the 2 types of the 2 kinds of pressure that drive fluid movement. So on the left, we can see the hydrostatic pressure. So due to fluid pressing against the boundary, like a capillary wall, the hydrostatic pressure pushes the fluid across the boundary. So in blood vessels, this is due to the blood pressure. The osmotic pressure, it's due to the non-diffusible solutes that cannot cross the boundary, and the osmotic pressure pulls the fluid across the boundary. So in blood vessels, this is due to the plasma proteins, So how do the pressures, the hydrostatic pressure and the osmotic pressure, drive the fluid across a capillary? So it's the net filtration that occurs at the arteriolar end of a capillary. So here on the left we see the capillary lumen, and in blue is the interstitial fluid. So the hydrostatic pressure in the capillary pushes the fluid out of the capillary, and that's about 35 millimeters of mercury, and the osmotic pressure in the capillary due to the presence of the plasma proteins pulls the fluid back into the capillary. That's 26 millimeters of mercury. So the net filtration pressure is 10 millimeters of mercury. So that's the net outward pressure. So as a result, fluid moves from the capillary into the interstitial space. So this figure here, we're looking at, so net reabsorption occurs at the venous end of a capillary. So the hydrostatic pressure in the capillary pushes fluid out of the capillary. The pressure here has dropped because of resistance encountered along the capillaries. So this pressure is 17 millimeters of mercury. And then there's the osmotic pressure in the capillary that will pull fluid into the capillary, back into the capillary, right? And we said again that it has to do with the presence of plasma proteins. And so this net filtration pressure is negative because it's a negative number at the venous end, and this means that reabsorption, not filtration, is occurring, and so fluid moves from the interstitial space into the capillary. That is a very key point. Homeostatic imbalance 19.2. Edema is an abnormal increase in the amount of interstitial fluid. It's caused by either an increase in the outward pressure, which drives fluid out of the capillaries, or a decrease in the inward pressure. An increase in capillary hydrostatic pressure accelerates fluid loss from the blood. So this could be a result of incompetent venous valves, localized blood vessel blockage, congestive heart failure, or high blood volume. An increase in interstitial fluid osmotic pressure can result from an inflammatory response. Inflammation increases the capillary permeability and allows proteins to leak into the interstitial fluid. And this causes large amounts of fluid to be pulled into the interstitial space. A decrease in capillary colloid osmotic pressure will hinder fluid return to the venous end of the blood, and this can be caused by hypoproteinemia. Okay, so that means that low levels of plasma proteins due to malnutrition, liver disease, or glomerulonephritis, which is loss of plasma proteins from the kidneys. Edema also can be caused by decreased drainage of interstitial fluid through lymphatic vessels that have been blocked by disease or surgically removed. Excess interstitial fluid in subcutaneous tissues generally causes pitting edema. Edema can impair tissue function as a result of increased distance for diffusion of gases, nutrients, and waste between the blood and the cells. Slow fluid losses can be compensated for by renal mechanisms, but rapid onset may have serious effects on circulation. Figure 19.20, this is called pitting edema. So applying pressure with a thumb leaves an indentation that remains for some time.